Hello, everyone, and thank you for tuning into my presentation. My name is Morgan Mark, and I'm a junior majoring in bioenvironmental engineering. And today I'm going to be discussing the results of my project where I assess the correlations between amphibian species of conservation concern. So just for a little background, an amphibian is any animal that usually starts off the beginning of its life in the water, then metamorphoses in a process that basically changes their entire physiology. And then they spend the majority or the rest of their life on land, only returning to the water to breed, escape from predators, or to find food. So that can be seen in the Greek root for amphibian, meaning living a double life, which is both in water and on land. And amphibians are especially important in ecology because just due to the nature of their physiology, their skin is extremely permeable to water since they tend to stay closely linked to that for the remainder of their lives. They are often very susceptible to various environmental chemicals and pollutants, which makes them great indicators for healthy ecosystems. So if there is a river that's very polluted, you usually won't find amphibians. And kind of the opposite of that trend, if you have a very pristine, clean ecosystem, you will usually find amphibians, which indicates that that healthy ecosystem is able to support a wide variety of life. And this is also important given the fact that a lot of amphibians, almost 50%, are threatened with extinction, which has been due to a variety of factors, including habitat loss, which is directly a result of human activity, as well as disease and pollution, amongst other factors. So it's extremely important to understand how amphibians are becoming extinct and kind of what drives those factors to better understand ecological health and to come up with mitigation strategies and conservation projects. So the way that we classify endangered species is according to the ICUN Red List status list. And that can be seen on the right over here, where least concern are animals that are extremely abundant and are not in any danger of facing extinction or being threatened. And as species numbers decline and there are more factors affecting their populations and their survival in the wild, they become more and more threatened and eventually can become extinct if their entire population is wiped out from planet Earth. So keeping in mind that over 40% of amphibians are threatened with extinction or threatened or will become extinct within a very short period of time, it's important to understand why this is happening and to see if there are any ways that we can kind of predict if a species is more prone to extinction and devise ways to protect these species in accordance with those parameters. So that was the rationale behind the question that I asked for this project is conservation status correlated with certain morphological factors? For instance, body length or lifespan or ability to lay eggs on land or in the water. And kind of an extension to that question is, can understanding these factors help in conserving these animals, therefore protecting the ecosystems and ecological services that they provide? So to begin this assessment, I decided to take a data set from Kaggle, which is an open source online platform where scientists, researchers, and hobbyists can upload data sets that they create themselves, and it's basically open to the entire community for anyone to use. So once I found my data set on Kaggle, which was entitled Predicting Amphibian Diversity, I read it into a pandas data frame. So as you can see in the picture on the right, this is what the data looked like when I downloaded it from Kaggle. And after I ran it through some Python processing, I converted it into a pandas data frame, which allowed me to be able to use this data and manipulate it accordingly. Some of the other things that I did to prepare my data was to clean the column names, so making them in a more readable format. For example, I converted ICUN status to another column that was just the short abbreviated version, which allowed for easier graph design and data analysis. And I also created new columns to represent values that weren't displayed in the original data set. So some parameters were given as minimum lifespan and maximum lifespan. So I decided to find the average of those two and create a new column that was average lifespan. In addition to processing the columns this way, I also used Seaborn for some preliminary plots to see if there were any overarching trends that I could discern from the data from the get-go and help me guide my analysis and see which factors might be correlated with amphibian status of threatened or endangered and their morphology. And finally, I used PyPlot to create the more specific plots for data analysis and to tell the story of the data that I discerned from this data set. So as I said, the first thing that I did was to create a Seaborn pair plot to basically visualize overarching trends that were present in the data. But before I could do that, I wanted to get a general sense of what this data was telling me and what was present in the numbers that were given. So as you can see from this table on the top right, I basically separated the data given into Anura and Chordata. Anura are frogs and Chordata are salamanders. So just to differentiate between those two groups of organisms. And I also separated them into their threatened and endangered status. So LC is least concern, NT near threatened, VU is vulnerable, EN is endangered, CR is critically endangered, and EW is extinct in the wild. And as you can see, there are more salamanders in this data set. So the data was slightly salamander skewed, which is important to consider as I explain my data. So with this pair plot, you can see that there are many factors that exhibit positive correlations. So basically what I decided to do was highlight 5 morphological parameters, specifically average age of maturity, body size, maximum lifespan, average offspring size, and average incubation period, which is the time that the eggs are allowed to develop before they hatch. And these just seemed like the 5 parameters that would most likely correlate with something like an endangered and threatened status. So when I plotted all of these parameters against each other, I noticed that there were some that had significant positive trends, which indicates to me that there might be a correlation between the two. In particular, you can see that mostly every parameter that is plotted against maximum lifespan exhibited a positive trend. There were others, but I decided to focus on this one just because each one showed a pretty clear positive trend, which basically indicates that there's a relationship between each of these parameters and the maximum lifespan that these amphibians usually live. To further examine the trend of maximum lifespan, I also created some bar plots to show that the median maximum lifespan seemed to decrease with more imperiled conservation status. So if you remember, least concerned is not threatened at all, where extinct in the wild is the maximum threat you can have and the organism no longer exists. So as you can see from the box plots in the bottom right, the median line, which is the middle line of the box plot, tends to shift to the left, which means the median max lifespan decreases as the species threat level increases. Additionally, I also wanted to plot some more factors, including max lifespan, in terms of the conservation status themselves. So after looking at everything holistically in terms of all amphibians and all conservation statuses, I decided to hone in on these specific factors. And since I found that maximum lifespan seemed to correlate positively with all of the other parameters I examined, I decided to start with that. And essentially what I did was I found the frequency of amphibians at each conservation status, and I plotted their median max lifespan as a bar chart. And as you can see from the color coding and just the results in general, the median max lifespan correlates very well with conservation status, meaning that least concerned generally had the highest median max lifespan, while extinct in the wild had the lowest. So although it's impossible to say that there's a definitive causation associated with a conservation status and median max lifespan, this data does suggest that shorter-lived amphibians are more likely to be endangered. And another parameter that I decided to model was median body size. And as you can see from the colors, aside from the fact that least concerned and non-threatened are slightly flip-flopped, it does also show that general trend where as conservation status becomes more threatened, median body size decreases, which suggests that smaller amphibians are more likely to be endangered. So when thinking about these two factors in terms of ecology and how this relates to endangered and threatened status, shorter-lived amphibians are more likely to be endangered. So that means that amphibians that don't have as long of a lifespan, maybe don't produce as many offspring, and they're just shorter-lived in general, probably are not able to deal with major environmental or like habitat disruptions that could really impact survival just because they live for shorter. So there's just more impact in general. Also, for body size, the smaller the amphibian, it might indicate that they are more likely to be endangered just by the fact that they're smaller, so they're more prone to disturbances, and they're not able to kind of mitigate those factors because of their small size. So looking at these two parameters in isolation, lifespan and body size were also positively correlated. So now that's looking at them in conjunction, as seen from the previous seaborne pair plot. So this could suggest that larger amphibians also live longer. So all three of these factors, median max lifespan, median body size, and conservation status, could all be very tightly linked. And these were the three factors that I found had the best positive correlations between conservation status and the parameters that make them more likely to be threatened. So I also decided to look at salamanders and frogs in isolation because all of the previous work I had been doing was for all of the amphibians in the data set. But I wanted to see if there was a difference between frogs and salamanders. So when I plotted the median max lifespan, but separated in this case for frogs and salamanders, I found that frogs generally had a lower median quantity than salamanders, which suggests that frogs are smaller, shorter-lived, and mature faster. So those were the other two parameters that I also decided to plot as box plots, but I did not show in this presentation. And I guess thinking about it logically, that almost makes a little bit of sense. Frogs tend to be smaller. Salamanders do tend to live longer in the wild than frogs. And that could indicate that frogs are more likely to be endangered than salamanders. So I decided to kind of test this hypothesis as well and create some pie charts that demonstrate the percentage of frogs and salamanders that fall within each threatened and endangered category. So as you can see, contrary to my previous hypothesis that more frogs would be endangered and threatened, a greater percentage of salamanders tend to be near threatened, vulnerable, and endangered, as seen by the darker blue portions of the pie compared to the least concerned, which is the light blue. So this indicated to me that it's not necessarily a holistic overall trend that I saw with all of the amphibians that applies to frogs and salamanders in isolation, which is interesting because that indicates that you need to really take these conservation studies and data sets on a case-by-case basis. And sometimes you can't just look at amphibians in total because it can differ for frogs and salamanders. As they are extremely different creatures, they tend to have different life histories and they might be impacted by different factors. And just for some more plots, when I examined frogs and salamanders in isolation, I created the same two plots as seen from 2 slides previously, where I looked at median body size and max lifespan for both frogs and salamanders in isolation. So as you can see with the salamanders, when you look at the median max lifespan, there's almost a reverse trend as what was seen for amphibians in general, where the least concerned species had the highest median max lifespan. But as you can see, they tend to have more on the shorter side. And the same almost holds a little bit true for the salamander median body size, where you can see that the endangered species tend to be closer to have a larger median body size than perhaps the vulnerable and the near threatened. So just showing that this is contrary to overall amphibian trends that I plotted previously, kind of supports the alternate hypothesis that I created that frogs and salamanders must be examined in isolation and that they don't hold true to the general amphibian trends. And overall, size and IUCN status correlate fairly well for salamanders, but not for frogs, which again is contrary to the overall amphibian trend that I found. just because, as you can see for the frog median body size, again, least concerned was the lowest median body size, but this was contrary to the overall amphibian trends that I found. And kind of to touch again on what this data means is that IUCN status seemed to be correlated with maximum lifespan and body size, but only when you look at all amphibians in total. So this would tend to suggest that endangered amphibians are really smaller and shorter lived, which ecologically and common sense wise seems to make sense. But these trends do not hold true when you only look at frogs or salamanders in isolation. So as you saw from the previous slide, there were opposite trends to the overall amphibian trend seen with both salamanders and frogs. So it's important to determine which species of conservation concern you want to try to protect, because that is going to determine conservation mitigation strategies. And two, making sure that you have the data that you need, because if you are trying to save salamanders and you instead have frog data, those those data sets might not correlate well, and you might ultimately end up with data that doesn't apply to the activity that you are trying to do. And finally, as we saw from the pie charts, more salamanders are threatened and endangered, but it must also be taken with the assumption and kind of the observation that the data set contained more information about salamanders than frogs. So it could have been skewed data. And this segues perfectly into my limitations of the study, was that correlation does not necessarily imply causation. So even though we found general overall trends for amphibians, and there was a seemingly very nice positive correlation between maximum lifespan and other morphological parameters, there are many, many, many other factors that could affect amphibian survival, reproduction, and ultimately conservation status. As I mentioned on the first slide, human activities are one of the biggest factors, which can be limited to, or limited but not excluding, habitat loss, pollution, and disease dispersal, which has really impacted amphibians over the past half century. So just because I decided to look at morphology doesn't mean that this is the only factor that really determines habitat, or that determines conservation status. If anything, it may not even correlate, and there could just be it may not causate, but it may just correlate, which is a good indicator, but not necessarily a proxy for conservation status. And as I mentioned previously, this data set was extremely limited, and it was only a small fraction of morphological factors tested, and maybe even a small factor of amphibians that are present over the entire world. So there are many factors that I haven't examined and may actually play a more critical role in morphological factor and IUCN status correlation. And again, as I mentioned previously, all species are unique. Even within frogs and salamanders, they cannot be treated the same. So when you are trying to come up with conservation strategies, it's best to examine the situation that you have at hand and to make sure you have the adequate data to truly come up with the best strategies, because trying to extrapolate data from a totally different data set, or even looking at amphibians overall, might not yield an accurate representation of the data that you need. And finally, this was kind of a static data set that just listed values for all of these parameters, but we don't know how any of these factors will change over time, how human activities will change over time, and therefore how species threatened in conservation status will change over time. So there are many uncertainties left to examine and for further study, but I think that this analysis proved a good baseline for examining what morphological factors could impact and correlate with IUCN endangered and threatened status. And with that, I would like to thank you for tuning into my presentation. I hope you enjoyed my talk, and thank you again.